Hello everyone, welcome to another session of online discussion. Here today we will continue with the respiration in plants chapter. In the previous session, we had a discussion on what is respiration, what is the major difference between respiration, breathing and combustion, and we also discussed what are the types of respiration, that is respiration can be aerobic as well as anaerobic. We also had a discussion on what are the different steps in aerobic respiration and what is the other name for anaerobic respiration. Anaerobic respiration, as we have seen, it is also known as fermentation, although when we talk about a minute difference between the term anaerobic respiration and fermentation, because respiration, when we talk about respiration, is something which is a part of metabolic activity of a cell, right? So anaerobic respiration, the term when we talk about it, is an intracellular process, whereas when we talk about fermentation, although the process that is happening during fermentation is an anaerobic respiration, but literally when we talk about fermentation, since fermentation is a commercial process, it is happening in a commercial vessel, there the term that is more correctly describing the fermentation process, it is an extracellular process. So anaerobic respiration is intracellular. The term fermentation will more accurately be defined as an extracellular process, because whatever process is happening during fermentation, all the enzyme that is produced, it is released outside, and whatever changes or the reactions are happening, it is basically outside in the medium, right? So that is the basic difference between these terms that we come across in the chapter. So now, as I was telling you, this aerobic respiration is a step-by-step process, right? So basically we have 4 steps, starting from glycolysis, link reaction, Krebs cycle, and finally electron transport system. Now also one more thing, aerobic respiration is a process wherein you can see complete oxidation or complete breakdown of glucose. So when the complete breakdown of glucose happens, it ultimately releases carbon dioxide and water, right? So that is how the complete breakdown of glucose will give you. Now in the process, after the glucose is completely broken down, all the energy that was stored in glucose can be released out, and this energy can be used to convert ADP into ATP. That is how maximum number of ATP can be harvested during aerobic respiration. When we talk about anaerobic respiration, it is the process where complete breakdown or complete conversion of glucose into carbon dioxide and water doesn't happen there, right? So that is the reason only partial energy can be extracted in case of anaerobic respiration. That's the reason we can see that the number of ATP that is produced during aerobic respiration and the number of ATPs that are produced during anaerobic respiration, there is a huge difference between the number, right? Like for example, in aerobic respiration, we will get around 38 ATPs, whereas during anaerobic respiration, or most, one more term that we can use there is fermentation, we only get around 2 ATP. So that is the difference between the ATP generation of aerobic and anaerobic. What is the reason for that? Because aerobic respiration is a process where complete breakdown of the substrate will happen. In this case, we are talking about glucose, and anaerobic respiration is a process of partial breakdown of the substrate, right? So now we will just go and start the process of aerobic respiration. So we'll discuss what are the steps that are involved in aerobic respiration, right? So here, aerobic respiration. Right? So in aerobic respiration, today the concept that we are going to see here is glycolysis, right? Which can also be called as EMP pathway, right? So glycolysis, which is also known as EMP pathway, based on the name of the scientist who discovered or elucidated or found out the pathway, Embeden, Meyerhoff and Parnas pathway. So glycolysis is a very important pathway for both aerobic as well as anaerobic respiration, right? So it is a slow step-by-step breakdown of glucose. So around about 10 steps are there in glycolysis. So before we go into the reaction, before we go into the metabolic pathway, let us see some introductory terms or introductory concept or introductory points that will help us to understand glycolysis in a better way. And all these points that I am going to discuss here is very important in terms of, competitive exam, because it is one of the important chapter for the competitive exam, as I told you before also. So let us see here. Now here, as you can see, glycolysis. So we will just split the term here, right? So when we talk about glycolysis here, it is comprising two words, glyco and lysis, right? So what do we mean by glyco? Glyco literally translates into sugar. Lysis means breakdown. Right? So simply put forward, glycolysis is breakdown of sugar, right? So breakdown of sugar to give energy, right? So now when we talk about this breakdown, it is not a complete breakdown. That means when we talk about glycolysis, it is a partial breakdown. Right? So this is a very important thing that we should remember. Glycolysis, the complete breakdown of glucose doesn't happen. Here only partial breakdown of glucose happens. Let me show you why it is partial breakdown, right? Because when we talk about glycolysis, right? So glycolysis, the substrate in glycolysis is sugar, that is glucose. Right? And the product of glycolysis is pyruvic acid. Right? Now you see here, glucose means it is having 6 carbon. So glucose contains 6 carbon and pyruvic acid is made up of 3 carbon. So you can literally see here a 6 carbon compound is broken down into 3 carbon compound, right? That means still you have 3 carbon or 2 bonds pending which can be holding some energy, right? That is the reason I am telling you it is not a complete breakdown, it is a partial breakdown, right? So what is happening here? One molecule of 6 carbon compound is broken down into 2 molecule of 3 carbon compound with the release of energy, but this energy is not the complete energy that is stored in glucose, right? So that is the reason I am using the word, it's a partial breakdown of substrate, right? So all these are important things. Now another important thing that is, many a times we get confused here is, since I am talking about aerobic respiration, many of us assume that glycolysis is an aerobic pathway, right? Because we think like, if in aerobic respiration, how can something be anaerobic, right? So that is the reason here. This point is very, very important here. When we talk about glycolysis, the next important point here, it is an anaerobic process. Right? So why it is called as anaerobic process? Because there is no direct involvement of oxygen, right? So why it is called as anaerobic process? No direct oxygen involvement. See here, I have used a term, no direct oxygen involvement, because when we talk about aerobic respiration, it is understood that aerobic respiration is a process that occurs in the presence of oxygen, right? Now we have aerobic respiration divided into 4 steps, right? So oxygen doesn't play a part in all of the 4 steps, right? So ultimate role of the oxygen comes only during electron transport system. So when we talk about glycolysis here, although it is a part of aerobic respiration, the pathway or the process itself is a type of anaerobic process, right? Now, another important question here, another important point here, it is a common pathway, right? So it is a common pathway for both aerobic and anaerobic respiration. right? So whether it is an aerobic respiration that we are talking about or anaerobic respiration we are talking about, glycolysis is an important part of both aerobic as well as anaerobic respiration. That is the reason I am saying here it's a common pathway for both aerobic as well as anaerobic respiration, right? Now the next important point here, right? It has oxidation step involved. Right? See, two contradicting statement here. I am saying it's an anaerobic process. So, complete when we talk about a pathway, it is an anaerobic pathway, right? But here there is a process or there is a step involved in the process. It's a 10-step reaction. So, one of the step in the process is an oxidizable step or oxidation step, right? When I say oxidation step, that is oxidizing agent involved, right? So there is a step here which involves the presence of oxidizing agent, right? So which oxidizing agent is involved here? NAD plus, right? So this is the oxidizing agent that is involved here. That is the reason I am saying or I am telling you that one of the step in the process is oxidation step, right? Now the next important point here, when we talk about glycolysis, it is a 10-step reaction, right? So the entire pathway, how many steps are there? It is a 10-step reaction, right? Now this 10-step reaction can be categorized into two phases, right? What are the two phases present in this reaction? Preparatory phase and payoff phase, right? So the whole of glycolytic pathway can be divided into two steps, preparatory step and payoff phase, right? So preparatory phase here, it uses ATP, remember this, and payoff phase here, it releases ATP, right? So that is the difference between preparatory phase and payoff phase, right? So these are few important points that we are discussing here, right? So it's a common pathway, it's a 10-step reaction, it is an anaerobic process, it's a partial breakdown which is divided into two phases, that is preparatory phase and payoff phase, right? So now we will talk about the pathway, how it happens. But before that, there is one more important thing, the site, right? So this is something very important where actually this glycolysis is happening in a cell, right? So when we talk about the site here, in prokaryotes and eukaryote, both type of cell, this particular process occurs in the cytoplasm, right? So what is the site of glycolysis in both prokaryote as well as eukaryotic cell? Cytoplasm. So since we are talking about respiration in plants, so definitely plant cell is an example of a eukaryotic cell. So in plant cell, where is that initial step of aerobic respiration happening? It is happening in the cytoplasm, right? So now before we go into the process, we should know that what kind of substrate primarily enters into the glycolytic pathway in plants, right? So there is something we should remember here. In plants, right, when we talk about the different types of substrates that are present which can be used in the respiratory pathway, all the biomolecules can be used. All means plant can use carbohydrate, it can use lipid, it can use a protein, right? But then lipid, protein are not a primary substrate for respiration. It is only used when the carbohydrate is missing or carbohydrate is not present, right? So now when we talk about plants, in plants, right, produced food is always in the form of which compound? Glucose, right? So what is the food that is produced in the plant? The food that is produced in the plant is in the form of glucose, that you also call it as photosynthate, right? And next, right, stored food. So stored food in the plant is in the form of starch, right? Similarly, transported food. So in which form the food is transported, right? So food is transported form of food. So the transported form of food is sucrose, right? So it is mentioned in your textbook here. Sucrose is the substrate that is actually going to enter into the glycolytic pathway, but sucrose cannot directly enter into the glycolytic pathway. As you all know that sucrose is a disaccharide, right? So it is still not a simple sugar, right? So it has to be broken down into the simple form, right? So what the plant will do before it undergoes glycolysis, the sucrose has to be broken down. So what they will do? They will break down sucrose. So in the phloem, food is transported, or the product of photosynthesis is transported in the form of sucrose, right? And that will be unloaded to each and every tissue, because what is the function of phloem? Phloem helps in transport of food, because we know that food is synthesized only in the leaf, green part of the plant, right? Leaves primarily, or the green part of the plant. It can also be extracted from the storage organs, but primarily leaf is the part from where the food is produced and distributed to different parts of the plant. So it distributes the food to different part of the plant with the help of phloem tissue. So phloem is one of the important transport complex tissues in plant, right? So what kind of food is getting transported through phloem? So the type of the food that is getting transported through phloem is not glucose. It is in the form of sucrose, which is a disaccharide, right? So now when this from the phloem, this food gets unloaded into the tissues, right? Why it is getting unloaded into the tissues? Because the food is required by each and every cell of the plant, so that it can make this food to undergo respiration, and whatever energy is stored in the food, they can extract that energy. But this food is in the form of sucrose, which has to be further broken down into simpler forms. So what is the simpler forms it will break down into? We know that sucrose here is a disaccharide, right? So it will be broken down into its unit, that is glucose and fructose, right? You know that both are monosaccharides. So these are simple sugars. So sucrose is broken down into fructose and glucose, and we know that glucose and fructose are isomers of each other, right? So now what will happen? Sucrose will be broken down into glucose, and this glucose will enter into respiratory pathway, right? So this is the preparation that the cell should do before the food enters, or before the respiratory substrate enter into the respiratory pathway. So glucose cannot directly, start with glycolysis. Sucrose cannot directly start with glycolysis. It has to first convert into glucose and fructose, primarily glucose, and then only the substrate or the most preferred form of sugar for glycolysis is glucose, right? So once this is done, now it is mentioned in the textbook, this particular reaction is catalyzed by the enzyme called as invertase, right? So this is the function of invertase, right? Now if starch also, the stored food also, if the plant has to use, the starch also has to be converted to glucose. The plant cannot directly take starch and start the process of aerobic respiration. First it has to be broken down. You know that the basic monomer of starch is glucose. So starch is also made up of different repeating unit of glucose, like amylose and amylopectin. These are the two subunits of starch. Amylose and amylopectin are individually also made up of repeating units of glucose, right? So that digestion, primary digestion, is very important before the substrate will undergo the process of respiration. reaction, right? So now this is the general introduction with respect to the glycolysis, right? So next we will talk about what are the different steps in the process of glycolysis. So now we will talk about the glycolytic pathway. So we will see the process or the pathway of glycolysis, right? Always remember it's a 10-step reaction. So if you ask me, sometimes the students have a, a question, do we need to remember all the enzyme that is involved in all the 10 steps, right? As per for objective exam, there are 3 important enzymes that we definitely has to remember in glycolysis. So anyhow, I'll write all the 10 enzyme here. So it is better you should have an idea, because all the enzyme, we have enzyme part in biomolecules wherein already we have studied the different types of enzyme and the classification of enzyme. So that will not be a hard part. So always remember every topic or every chapter has some connection to other chapter. So it's like a sort of revision. So if you remember the enzymes of all the 10 steps, you can recall from some other chapter where you have studied all those enzymes. So it is actually a sort of revision, right? So now we will talk about what are the different steps and what are the substrate product, what are the byproducts of these steps, and ultimately what is produced during the process of glycolysis, right? So as just now we discussed, sucrose is converted into glucose and fructose. Fructose can also be converted back to glucose because glucose and fructose are isomers of each other. Both are 6-carbon sugar. It is monosaccharide, which you also call it as hexoses because it is made up of 6 carbon, right? So now first, as I told you, the entire glycolysis can be described into 2 phases, preparatory phase and payoff phase, right? So we will talk about the first phase here, which is called as preparatory phase, right? So I will start with the substrate, right? So what is the substrate that enters into the glycolytic pathway? Glucose. So glycolysis can also be called as breakdown of glucose. Since I already told you, breakdown of glucose here is not complete, it is partial breakdown. Glucose is being broken down into pyruvic acid, right? So glucose, one molecule of glucose which is made up of 6 carbon, right? So glucose, so if you, the one molecule, we will talk about it later. So 6 carbon. So glucose contains how many carbon? It's an hexose sugar. It is made up of 6 carbon, right? So as soon as glucose enters into the cell, the first step that happens here is glucose is converted to glucose 6-phosphate, right? As you can see here, what is the difference here? Glucose and glucose 6-phosphate. So what is added here? This phosphate group is added at the 6th carbon, right? So at the 6th carbon of glucose, a phosphate group is added. So who is donating the phosphate group? The phosphate group is donated by ADATP, right? So it is donated by ATP, and in the process ATP is converted to ADP, right? So this is the step where energy is not getting produced, rather energy is used in the process, right? So glucose in the presence of ATP is converted to glucose 6-phosphate. So this is the first step here. It is catalyzed by a very important enzyme called as hexokinase, right? Remember, this is an irreversible step. So this step is not reversible, and this enzyme is also subjected to feedback inhibition. So this step can also be given as an example of feedback inhibition, because if the substrate is, the product accumulates more, ultimately it will inhibit the enzyme, right? So I was telling you, out of 10 steps or 10 enzymes, there are 3 enzymes which is very, very important for the process. So one of the 3 enzyme is hexokinase. So the first enzyme which is important for the process of glycolysis here is hexokinase, right? And the cofactor for hexokinase is magnesium. So magnesium is a cofactor for hexokinase. So what kind of reaction is happening here? So it is addition of phosphate. Glucose is getting added, phosphate is getting added to glucose to convert it into glucose 6 phosphate. So what kind of reaction is happening here is phosphorylation, right? So the entire reaction can also be called as phosphorylation reaction, right? So next step, what will happen here? Glucose 6 phosphate is converted to fructose 6 phosphate, right? So see here, glucose is converted to fructose. Recently, just now I told you, glucose and fructose are isomers of each other. That means this step is a reversible step, right? Because both are isomers of each other, right? So now this step, no energy is used. It is simply rearrangement or isomerization reaction. So this is the second step here. It is catalyzed by the enzyme isomerase, right? Or phospho or hexoisomerase enzyme, right? So that is the second step here. Next step here, third step, right? This fructose 6 phosphate is converted to fructose 1,6 biphosphate, right? You can see here, fructose 6 phosphate is converted to fructose 1,6 biphosphate. Here it was only fructose 6 phosphate, only at the 6th carbon. Here a new phosphate group is added at the 1st carbon. So 2 phosphate group is added. So new phosphate is donated by whom? Again, the new phosphate is donated by ATP, right? So it donates the new phosphate and gets converted to ADP. Right? Now this is a very important step here. This occurs in the presence of an enzyme, phosphofructokinase. Right? So what is the enzyme here? Phosphofructokinase. See here, kinase. Here also kinase, right? So the type of reaction here also is phosphorylation, right? So the third step also is phosphorylation, right? So as I was telling you, the 3 most important enzymes. The first one is hexokinase. The second important enzyme here is phosphofructokinase, right? So one very, very important information with respect to this enzyme, that is phosphofructokinase. It is a rate limiting enzyme, right? So rate limiting enzyme, right? So there are many questions related to this that you can see in multiple sources. Which step will decide the pace or the rate or the speed of glycolysis? It is decided by this particular enzyme, which you call as phosphofructokinase. So what is the rate limiting step in glycolysis? When glucose is converted, fructose 6 phosphate is converted to fructose 1,6 biphosphate, right? Ultimately, what will happen? See all these, right? So glucose is 6 carbon, glucose 6 phosphate is 6 carbon, fructose 6 phosphate is 6 carbon, fructose 1,6 biphosphate is also 6 carbon. Ultimately, what will happen? The fructose 1,6 biphosphate will split up, right? So the next is splitting up of fructose 1,6 biphosphate. So fructose 1,6 biphosphate will split up. So this is your fourth step, splitting up, right? This is known as aldol condensation. So that is the reason the enzyme here it is aldolase, right? So what is the enzyme that is used in this process? Aldolase, right? So aldolase is the enzyme that is used for the splitting of fructose 1,6 biphosphate into phosphoglyceraldehyde, PGAL, or you say glyceraldehyde 3-phosphate, PGAL. You can also call it as G3P, glyceraldehyde 3-phosphate. Another compound that is formed here is DHAP, dihydroxyacetone phosphate. So what is happening here? 6-carbon fructose is being split into 2 molecules, right? So 6-carbon fructose is split into 2 molecules. What are the 3 molecules here? We will see. One 3-carbon phosphoglyceraldehyde, another 3-carbon dihydroxyacetone phosphate, right? So these 2 molecules are produced as a result of splitting of fructose 1,6 biphosphate, right? So 6-carbon is split into 2 3-carbon molecule. So here remember, these two particularly, they are isomers of each other, right? That means they can get interconverted, right? So here, this is our 5th step. Again, what is the enzyme? Isomerase, right? So what is the enzyme here? Isomerase enzyme, right? So this is our next step. Now what will happen? DHAP will also be converted into PGAL, because the continuation of the process, for that purpose, DHAP will also change to PGAL, and then it will continue forward in the glycolytic pathway. So this particular phase that you are seeing here, from glucose to the splitting of fructose 1,6 biphosphate, this whole process here, from here to here, it is known as preparatory phase, right? So you can see here, it is called as preparatory phase. As you can see here, in preparatory phase, there is no ATP released, right? On the contrary, ATP is being used, right? So let us see where the ATP is being used, right? You can see here, here is one ATP that is used when glucose is getting converted to glucose 6-phosphate. The second ATP is being used when fructose 6-phosphate is converted into fructose 1,6 biphosphate. So in preparatory phase, right, ATP is used. How many ATP is used? 2 ATP is used, right? And what is produced here? The 2 3-carbon molecule, that is PGAL and DHAP, right? So let us see what happens here. Right? So now this G3P and PGAL, right? So it will go forward to complete the process, right? So let us see how it completes the process, right? So now what will happen here, as DHAP forms G3P, this is converted into, right? So remember, PGAL and DHAP, there is one more name that we use for it. This too is also known as triose phosphate, right? So what do you mean by triose phosphate? 3-carbon sugars are also known as triose phosphate, right? So now what will happen? This DHAP and this, they will combine, right? So when they combine here, this process that is happening, combination of DHAP and G3P, this is your 6th step, right? Here it is triose phosphate dehydrogenase enzyme, right? So what is the enzyme used here? Triose phosphate dehydrogenase enzyme here, right? So now what will happen? This will combine. So this is our 6th step. using triose phosphate dehydrogenase. Now this is the step what I was telling you as an oxidation step, right? So oxidation means what is the oxidizing agent present in the cell? NAD plus, right? Which is converted to NADH plus H. Right? So this is the oxidation step of glycolysis. So this is the sixth step catalyzed by triose phosphate dehydrogenase enzyme. So wherever this dehydrogenase term you come across any such enzyme, it participates in the process of redox reaction, right? So here what is the product? The product is 1,3-phosphoglyceric acid. 1,3-PGA is the product. 1,3-phosphoglyceric acid. Remember, these are the two 3-carbon compounds. This is also 3-carbon compound. So how many molecule it should be present? 2 molecule, then only it can be equal to the 6 carbon of the glucose, because ultimately this bi-PGA is derived from the glucose, right? So glucose was 6 carbon. Ultimately till here when we talk about, it has converted into 1,3-biphosphoglyceric acid, which is a 3-carbon compound. Now 3 carbon, 3 into say 2, it will make 6 carbon glucose, right? So now one more important thing which can ask in the form of question. This step also involves the presence of inorganic phosphate. That is the reason. See here, we have 1,3-biphosphoglyceric acid. So it also requires an inorganic phosphate molecule. Remember here, since it is 2 3-carbon molecule, PGAL also is 3 carbon, DHAP is also 3 carbon. So how many NAD is required? 2 NAD. And how many NADH plus H is formed? 2 NADH plus H is formed, right? So 1,3-biphosphoglyceric acid is converted to 3-phosphoglyceric acid, right? So what is happening here? 1,3-biphosphoglyceric acid is converted into 3-phosphoglyceric acid. So what is the difference here? Here you had biphosphate, 2 phosphate was there, one in first carbon and third carbon. In this product you have only 1 phosphate group. So that first carbon phosphate group is being given out, right? So now who is accepting the first carbon phosphate group? ADP will accept this phosphate, gets converted into ATP. Since it is 2 molecule, right? So we have 2 ADP and 2 ATP. Now this also is a 3 carbon because it is derived from here. So here also it will be 2 molecule, right? Now this enzyme, this particular step is the 7th step. It is phosphoglycerokinase, right? So phosphoglycerate kinase, right? Next, what will happen? This 3-PGA will be converted to 2-PGA, right? It's not like 3-phosphate group is there. Third carbon, you have phosphate group. It is converted to second carbon, which you have the phosphate group is being shifted to second carbon. So it is just a rearrangement. From the third carbon, phosphate group is shifted to second carbon, right? So here also 2-PGA means it is also 3 carbon. So this was 2 molecule. This should also be 2 molecule, right? So this is the 8th step. It is just a rearrangement reaction. So now since it is a rearrangement reaction, right? The enzyme here is mutase. Phosphoglycerate mutase is the enzyme. Remember, this is a sort of rearrangement, right? This is again phosphorylation, right? What kind of phosphorylation? Substrate level phosphorylation. This is something very important you should remember. What is the first substrate level phosphorylation happening in glycolysis? When 1,3-biphosphoglyceric acid is converted to 3-phosphoglyceric acid. We will discuss what do we mean by substrate level phosphorylation later, right? So now this 2-PGA is converted to phosphoenol pyruvate, right? So 2-PGA is converted into phosphoenol pyruvate. Phosphoenol pyruvate again is a 3-carbon compound. How many molecule it should form? 2 molecule it should form, right? So this is your 9th step, catalyzed by the enzyme enolase, right? Now another important step here, question here, it's also a dehydration step, right? So what is happening here? Dehydration, removal of water molecule. So sometimes they may ask you, what is the dehydration? Where is this dehydration happening in glycolysis, right? So you should remember in the 9th step where 2-PGA is converted into phosphoenol pyruvic acid, right? So now let us see here what is next, right? So now finally this phosphoenol pyruvic acid will be converted into pyruvic acid, right? So pyruvic acid again is a 3-carbon compound, right? How many molecule it should form? 2 molecule, like here also 2. So here also 2 molecule, because we are talking about from 1 glucose. So 1 glucose is 6 carbon. So when a 3-carbon molecule is formed from 1 glucose, so how many 3-carbon molecule can be formed from 1 glucose? 2 3-carbon molecule can be formed. That is what this 2 I have written here, right? So 2 molecule of pyruvic acid. So what is the product of glycolysis? The product of glycolysis is 2 molecules of pyruvic acid. So phosphoenol pyruvate and pyruvate or pyruvic acid. Remember, pyruvic acid, sometimes you may also get here like pyruvate. Remember, this is acid and this is base, right? So this is acid, this is base. Now here is the another step where your one more phosphate is released, phosphoenol pyruvic acid. So this phosphate is not present here, it is simply pyruvic acid. So that phosphate group which is donated or left from phosphoenol pyruvate is again accepted by ADP to convert into ATP. So again it is 2 molecule, 2 ADP and 2 ATP. So now this is your 10th step, which is catalyzed by enzyme pyruvate kinase. So as I was talking about 3 important enzymes for glycolysis. So that is the 3rd important enzyme which is participating in the process of glycolysis. Although all the 10 enzymes I wrote, right? So if you remember this 3 enzymes, it will be very helpful for you to answering most of the question related to the enzymatic part of this process, right? So this is the 3rd and the final enzyme that is important or play a major role in the process of guiding, regulating the process of glycolysis, right? So this is pyruvate kinase. So this process, what we have just seen, this phase here is known as payoff phase, right? So in the payoff phase when we talk about, right? So let us see how many ATPs are used in the payoff phase, right? So in the payoff phase here, payoff phase, right? This 2 ATP is released and this 2 ATP is released, right? So when we talk about payoff phase from here, right? So payoff phase, ATP is released. How many ATP is released? 2 ATP is released, right? So 4 ATP is released, 2 here and 2 above, right? So ATP, number of ATP released is 4 ATP is released, right? So now if I want to find out what is the ultimate gain of ATP, so here net gain of ATP. So when I say net gain of ATP, I am referring to direct ATP or ATP that is produced in substrate level phosphorylation. It is 4 released, 2 used. So how many ATP is the net gain? 2 ATP. Remember, This 2-ATP, I am just referring to the glycolytic pathway, not the glycolytic pathway through aerobic respiration. In simple glycolysis, how many directly produced ATP you get? It is only 2. So net gain of ATP in glycolysis is 2. If there is no word like ETS or aerobic respiration, we have to go with the number 2-ATP, right? So that is all about our glycolysis. And what are the, so what the student perspective, from the student's perspective, what you need to remember here, this is a very important part, right? Next, as what are the steps that are using ATP, what are the steps that are releasing ATP. When I say what are the steps, you should know what is the substrate, what is the product. Like for example, one of the steps that is releasing ATP is when PEP is converted to pyruvic acid. So you should remember this reaction, right? And as I told you, 3 enzymes, hexokinase, phosphofructokinase and pyruvate kinase. So these are the 3 enzymes that decides the overall process or metabolism of glycolysis. In that, the enzyme that decide the speed and rate of glycolysis is your phosphofructokinase, right? So we'll stop here for today. All this again we will discuss when we talk about the balance sheet of respiration. Thanks.